everyone, and welcome to today's episode of Seed Speaks. My name is Ashley Robinson, and I'm the editor of the Alberta Seed Guide. Today, I'll be acting as your host for this episode. On today's episode of Seed Speaks, we're taking a closer look at how plant breeders are considering allergies, and food intolerances when it comes to plant breeding. And we have a few guests here today to discuss this with us. Thank you to Olof Olsson and Sachin Rustgi for joining us. And to those watching, please don't hesitate to ask any questions you may have for our guests in the chat box. And we're going to start by getting to know our panelists. So, Olaf, if you can please tell me about your research work and what the ScanOats Industrial Research Center is.
Yes, the ScanOats Research Center is a program that's focused on oats. So we work on different aspects on oats, all the way from the oat genome to develop new oat varieties, to enhance agronomy on the oat varieties we grow, look at important health promoting compounds that these oats could produce. And we also look at a little bit fermentation, scaling up processes. And so this is sort of different work packages, but we're all united around oats. And of course, in today's aspect, when it comes to genomics and when it comes to what's in the oat genome related to health, is of course relevant. And my own research is now focusing on oats, but also on wheat and quality compounds in wheat, but also agricultural properties like salt tolerance and nitrogen uptake efficiency and these kind of things. So I work over quite wide area.
Thank you, Olaf. And of course, we have Sachin. Thank you so much for being here today. And I know you work a bit more on the wheat side of things with your research work at Clemson University. So can you please tell me a bit about that?
Sure. Thank you so much for inviting me for this. I work on the development of reduced immunogenicity wheat lines, and we have two different streams of research that are going in this direction. So we work in eliminating wheat gluten proteins, especially which are immunogenic. So we were not working on developing zero gluten wheat lines, but developing wheat lines which were reduced in the content of immunogenic gluten proteins, which are generally gliadins and low molecular weight glutenins. And there were very few cases that were reported for the high molecular weight glutenins to cause any sort of immune reactions in the individuals. And I would like to also mention these high molecular weight glutenins are also responsible for the baking properties largely. So I know the baking properties are pretty complicated, and both kind of proteins, gliadins, glutenins, do contribute, but largely you need the high molecular weight glutenins to actually bake a loaf of bread. The other thing that we do is the gluten detoxification. So we also work on introducing these glutenases or gluten degrading enzymes into the wheat grains, which only express in a very specific conditions which require acidic pH. So these enzymes are selected such they will not process these proteins while the grain is developing or a dough is being kneaded or something so it will be actually expressing in a very acidic pH first. So I can elaborate on these things later on in the talk. So that's what we largely do. And lately we have actually start developing multi-gene editing, using multi-gene editing approach to develop the mutants in the gluten proteins, so we could actually have some lines which were mutants rather than actually transgenic in origin. And we have also screened a large collection of wheat lines from the mutants that were collected in early 1970s from the high altitude regions of the world, and they carry mutations in these gliadins and glutenins. So that's what largely we do.
Thanks so much, Sachin. You guys both are doing some really interesting work, and we're going to have a great conversation. So now let's start on those questions. So Olaf, my first question is for you, as I know ScanOats was involved in the recent decoding of the oat genome. And how did this help researchers understand why oats seem to provoke fewer allergies than some other cereal crop varieties?
Yes, right. So then once we had assembled the whole oat genome, which is a tremendous task because it's a very big one, it's also rearranged and those things. But then we could start looking at gluten prolamina-like proteins, they have to call it, which is also where glutens are involved. These are around in many plant systems, like in barley and in rye, for instance, and also in oat where you have the avenines. So what we did, we scanned through the whole oat genome for gene sequences that looked like something that was correlated to gluten or gluten-like proteins. Of course, we found many, and because it's a great homology between the systems. So this is the first part, but then we noticed that there was some lack of some. If you look at glutens itself, it's glutenins and gliadins, as we just heard, and there are many different classes, while some are more toxic than others. And there are special epitopes that are considered very toxic because they react with transamination reactions, and they are picked up by specific antigen-producing cells, and then they are interactive with T cells and cause a disease. So there are some targets one can look more specifically at. And we did it in collaboration with Angela Juhas in Australia, and where we also looked at, it's one thing that you have the oat sequences on the genome. It's a different thing which ones are actually expressed. And then we could see that from the beginning, avenins, it's a very small proportion of the oat storage proteins, in contrast to wheat, where the prolaminar proteins or glutens are the majority. So already there we have much fewer proteins in oat. And also when we looked at the most toxic ones, the alpha and the gamma ones, they are not present at all in the oat genome. But we have more the high molecular weight ones that we just heard that are considered less toxic. And also when you start looking at the ones that are left, we find some of these epitopes that are correlated to the celiac disease in some of the genes, but they are very much fewer in number, and they are also not the whole epitope, they are just parts of it, which means that they probably will not react in the same way with the antibodies like the gliadins would do. Which means that so on many different levels, from the presence of the genes to the expression of genes to the part of the epitopes to the structure of the epitopes, oat is different and is more favorable for celiac disease patients. So the conclusion from all this is that oat is most likely safe to eat for celiac patients, like we knew before, but we now have the molecular evidence that it should be like that. There are some oat varieties that could contain some of these epitopes, but in that case we could mutate them away quite easily, since we know exactly where they are and which they are. So, but it might not even be necessary because it's already so little there. So this is a very short summary of a very long story.
Yeah, and of course, you guys did some really important work, because I know, like, I have some friends that are celiac, and it's always that they can eat honey nut Cheerios because they're made from oats, which I always find really interesting. And of course, one of the big things with celiacs is that they can't eat wheat flour, which is of course in so many products. So how does the work that you guys were able to do on decoding the oat genome help with maybe being able to breed future varieties of other cereal crops such as wheat that would be able for celiacs to eat?
Yes, that of course one can use this knowledge, but this is a little bit of a circular argument, because we have used all knowledge from other systems to look for something that we think is poisonous and that we cannot find in oats, which means, and we know that oat is more or less okay for people with celiac disease, which means that we are probably right when we predict which proteins are dangerous or not. So the next step will be, like we just heard now, that we start looking at in wheat for the absence of these proteins and maybe enhance some of the good proteins, because you want to keep the baking properties at the same time. But the high molecular weight glutens, as we just heard, they are normally not very poisonous. And I think it's also possible to add on baking properties by increasing the level of some other fibers like arabinosilans that we also work with in ScanOats. And we have high arabinosilans oat, for instance, and this will enhance the baking properties of the oats, despite the fact that there's not so many gluten-like proteins in it. So man can also combine it in other ways and find other alternatives to gluten, or complement gluten with other alternatives. So I think the systems can learn from each other, but I think the general technology and everything is similar in wheat and oats. I think I think they know all this already, basically.
Definitely really interesting. And I like how you were mentioning that when you are playing with these genes and stuff, you always have to still make sure that you're keeping those properties that allow these crops to be able to be baked
Yeah,
into these goods
yeah,
because that's what we need them for.
exactly. So oat. I mean, oat flour doesn't. You don't have the same baking properties in oat as in wheat. It doesn't lift the same way, and it's because the gluten is different or the avenins are different, so they're much less. So you can see it already there. So what we want to do, of course, we want to increase baking properties in oat. But as I said, we do that in other ways than with the glutens. We do it with other fibers, basically.
Definitely. And I think this is a good lead into Sachin, my next question for you,
Mm.
which I know that you've actually been involved in breeding a wheat variety that people with celiac disease can eat. Can you tell me a bit more about it?
Yeah, as I mentioned earlier, like we were actually having two stream of researchers like that we were doing. Like so we had like two kind of beads created in our laboratory where we have actually targeted a master regulator of these gliadins and glutenins. So gluten, people have a misconception. It's not a single protein, as Olaf has also mentioned. Like it's a large collection of proteins. So there were about like 70 to 100 proteins in the wheat that are accumulating in the wheat grains, and these proteins are classified in different groups like high molecular weight glutenins, low molecular weight glutenins, and gliadins of various kinds. And gliadins, especially the alpha gliadins and gamma gliadins, are extremely toxic for the celiac patients, and omega gliadins are actually responsible for the wheat allergies. So these gliadins can be eliminated. So research has been done and it has been demonstrated. So one can reduce the content of these gliadins or completely eliminate them. It will still bake into a bread because these high molecular weight glutenins actually kind of serve as like these fibril network, and then these gliadins will serve as the glue between them. So the dough mixing properties do change when we actually remove these things. So we have removed the gliadins and it has actually made a cookie making wheat flour into a bread making wheat flour, so that has a strong mixing properties. So because we removed like that glue between these fibrils, so these high molecular weight glutenins that we were keeping, and so we killed this master regulator that I just earlier mentioned, like it's called Demeter, and it has eliminated these gliadins and low molecular weight glutenins because their promoters are actually methylated in endosperm, which is the seed storage, the component of the seed that accumulate these storage proteins. And if it's not demethylated, so these methyl groups are like the decorations on the DNA, like so if it's methylated, it is inaccessible for transcription or produce a transcript, and that transcript is, that is RNA, is actually coded into a protein. So you will eliminate the protein like if you check the transcription of that particular gene. So that's what we did. We have already lines created that were actually about 89% reduced in the gliadin content. We have also parallelly worked on developing these lines which were expressing glutenases. So we selected two enzymes. One is a endoprotease from barley. So this is a protein degrading enzyme. So it works on proteins. That's why it's called a protease, and it actually has a specific specificity for the glutamine residues. So you might be hearing this word prolamins. So these proteins are very rich in protein and glutamic acid. These are two amino acid residues, and when they are repeated in a protein sequence, it cannot be processed by human digestive system. So there are peptides that are left undigested and they get into the bloodstream, and in the people who have the leaky intestine syndrome or have the correct genetics or predisposed for celiac disease, it will actually lead to the celiac disease. Or in the people who are allergic, the immunoglobulin E like actually binds with it and have the allergy symptoms. So these are very different medically, very different conditions. So when we express these glutenases, they can actually process them at the right sites where the glutamine and proline residues are present. And why the protein might have these, the plant has it like is because it increased the packaging efficiency in the seeds, because you can accumulate like the glutamine has two nitrogen atoms and the proline creates a kink in the protein filament, so you can pack more nitrogen in the grain. So we we can actually express these proteins. Like the second protein we selected is from Flavobacterium meningosepticum, and that's a prolyl endopeptide. That means it cuts after the proline residues, so you can actually process these undigested pieces or epitopes into fine diamino acids or single amino acid residues, so it could be non-toxic afterwards. So these were the two things that we have created, but both are transgenic. So as I mentioned, we were also screening the large germplasm collection, so there are a lot of mutants that we have screened, and we have access to a mutant that is omega gliadin free. It was actually developed back in Poland. And there is another mutant that we received from Italy, like, and that has um, the alpha and beta gliadins very low in quantity, so they will be bred together, like to have a line that could have reduced omega gliadins as well as alpha beta gliadins. And we plan to knock out the gamma gliadins using the CRISPR approach in those lines. So this is how, like, we are creating the material that will be available for people to use. And one thing I would like to mention, like, is the earlier two approaches are transgenic. However, the transgenics today are not accepted largely in United States and many other parts of the world. But I would tell, like, I mean, the time is coming. Like, you can see the Argentina and Brazil has approved the production of a transgenic wheat line, which is drought tolerant, and hopefully, like, it will be the other parts of the world as well. I don't know, but that's what I wish to express. Thanks.
That's really interesting, and I think that launches into another question. You were kind of mentioning the breeding techniques you've been using with some of these wheat lines that you guys have been working on, and of course, we're seeing a lot of newer breeding technologies. And yeah, there is, if we're not going into politics, but there is, of course, the debate about whether to approve some of these, such as gene editing. So, how do you guys think these tools can be used to breed crop varieties that people with allergies and food intolerances can eat? And whoever feels like tackling this question first can go for it.
Yeah, I can start maybe. Yes, because it's a matter of defining what is allergenic. I mean, and find the compound and then find the corresponding gene, and then mutate that gene or knock it out if you want to do that in ways. And the best way to do it at the moment, when you look for regular things, is to do it by mutagenesis. And we have in oat and also in wheat, we have produced mutagenized population with many different mutations. So what we do, we just screen populations for lines that happens to have a mutation in the target gene we are interested in. So we don't put in a mutation ourselves, we let sort of the mutagen do it. And if you do it that way, it's considered not GMO, strangely enough. So then that's allowed to get around all the legislation and everything. Because of course, then there are more direct ways to, just by editing, you can go in and by editing, edit the same exactly the same place. And then it's sort of in a borderline between GMO and not GMO. I think editing is released. It's released a lot now. They can probably start using it. But there are some cases when you really want to do GMO, when you want to add some more genes in that are not in the genome. And I think it's also one good thing one can do. At one point, we also developed gluten blocking peptides. So we by phase display, I don't go into this very complicated technology. We isolated small peptides that will interact with the gluten epitopes on the site where the transamination occurs, which means you block this reaction and they become non-poisonous. And we show that in vitro, very nice experiments. And that would also be a way to add in such activities into the genome of wheat, for instance. And then you would block out some of these dangerous proteins by producing peptides in vivo. So there are many ways to do it if you can use all the genetic tools that are available to us. But in some cases, then we end up in legislation problems and public acceptance and all these things that would delay the technology and maybe sometimes even stop it. But I think that's a different discussion from what we have today. But we could talk a lot about that otherwise.
Yeah, definitely. Sachin, is there anything else you'd like to add to that?
Yeah, I think it was very nicely summarized by Olaf. Like, but yeah, I mean, the thing that's true, like, we can actually screen and then use like the mutants. So we, as I said, like, we were working on like the naturally existing mutants, as well as like these were the lines that were collected from the high altitude regions of the world, like especially from the Nepal, and they were naturally having like a lack of these toxic proteins. And I have no idea like why these lines do not accumulate them. But we were using them. Like, but as I mentioned, like these are 70 proteins about, like, so you cannot kill and stack all of these mutations together like in a doable time frame. Like, so the idea of like having say like the CRISPR approach used, or like use the transgenic approach, came in play because you can actually kill the master regulator, and this master regulator you cannot mutate because it's like a demethylase. So you can actually create some side effects, like especially in the pollen fertility reduce, like if you knock it out, like, so that we have performed. So we have created transgenic lines where you can actually only suppress the gene specifically in the endosperm, that is the part of the seed which accumulate these storage compounds. So that kind of things you can do like with the transgenic approach, and hopefully like the time will come, like we were just waiting for it. And as you are aware, like the breeding process takes time. So these new technologies can shorten the duration, especially the CRISPR approach. It will be still several years of testing. I don't say like it will be just cut it down like to say like a single year. It will still be 4 to 5 years at the very least, like to screen the materials and then test them in various ways, like, and then make them available to the public.
Thank you. Yeah, I think that's really important to keep in mind when we do talk about plant breeding, any kind of plant breeding, that it definitely isn't something that we can do overnight. This takes a lot of work, and you guys have been doing, I'm sure, have put years of research into being able to release and share this information that we're talking about today. So I think my next question is, of course, what other you guys are focusing a lot, of course, on people that have cereal intolerances, such as like celiac and stuff. So what other food intolerances or allergies do you think we can tackle through plant breeding? Because there are a lot of other things that I know people are affected by, and stop them from being able to eat certain foods.
I think I mean it's a matter of how much you know about things, because I think as soon as you know the molecular mechanism behind how this allergen is produced, then you can block it. Then you find the gene and you block it. So I think that's just a matter of taking gene by gene that produce something that could work as an allergen when you eat it, and make sure that activity is not there. And so the problem is to define the compound exactly and define the gene that express it. But in theory, you can block anything as soon as you know what it is, basically. So
Interesting. And Sachin, is there anything else you'd like to add?
Yeah, there are multiple grains like where we can actually tackle this issue. Like, so I'm already working on the peanut allergies. Like, so the peanut has a very similar problem. Like, you could be seriously allergic to these proteins, and there were 4 proteins that were identified in peanuts to be major allergens. So about like 90% of the US population are actually affected or sensitive to those 4 proteins. So we were actually screening the germplasm collection very similarly, identified already the lines that were actually lacking these, and we are making genetic crosses to stack them. So that is kind of the route like that is more acceptable, as has been said earlier. uh, in parallel, we were also using the genome editing approach to actually kill these 4 major targets in the peanut, so it will produce a reduced allergenicity lines. And one might think like how it could be used, like, so the idea there was like, I mean, one can actually reduce the allergen content, so the others who are not sensitive can actually use these lines in the public space, and one could actually use these lines to kind of train the immune system, so one can actually develop kind of resistance or tolerance toward these grains, like if the slow doses of these allergens were actually introduced in a medical setting. So that's where like similar sort of research would be done, like, and there is already research being done in soybean, like where people have knocked out these allergenic proteins. And I believe like maybe in the other crops like corn and rice, where the certain proteins were identified to cause allergy in specific populations, might also benefit from this research.
That's really interesting. I didn't realize you were involved in some peanut research, because I was trying to get someone on here actually to talk about peanuts, because peanuts are such a nuts are such a common allergen. So thanks so much for mentioning that and telling us about it. So I guess we're getting a bit closer to the end of our show. So maybe I'll just get you guys to talk a bit about what you guys are kind of building off your research and wanting to be able to do now. And I'll start with Sachin this time. If you can just explain a bit more about what you're looking at moving into the future with your research.
Yeah, the major things that we were doing, like, I mean, we were focusing on developing the multi-gene editing platform, and we have also kind of uh, focusing on like the delivery systems because, as Olaf has mentioned, like gene delivery is also an important component. If you're using agrobacterium biologistics approach, you will be still introducing the foreign DNA that will integrate into the plant genome. So we were trying to find technologies that could be used. So we were working on a viral delivery system where we can actually transiently express these CRISPR enzymes, which is the Cas9 or Cas12a, and it could actually mutate the cells. And these mutants will be kept, and the virus will disappear from the face. So that was the idea that we were pursuing, like at the moment. So we could actually create the mutants instead of the transgenic lines or GE over GM lines. That's what we were working on. And the other thing we were doing is like to collaborating with more people in medicine so they can actually appropriately test the materials that we have developed.
Thank you. And Olaf, your turn.
Yeah, and that's one of the big future things, I think, when it comes to plant breeding is also to come into preventive medicine. I mean, to make sort of the food, you make food so healthy, so you don't need medicines 5 years later. So you sort of built into the food what you eat, something that will prevent, for instance, sugar peaks or insulin response and these things if you're diabetic, and you make it much more healthy than we have today in a scientific way. So everybody talks about what is healthy and what is not healthy, but one could do a lot of science and actually produce wheat or oat or anything that really will stop disease. I mean, oat already has beta glucans at lower cholesterol levels, and we are now working a lot in wheat to produce a very big complex in wheat that will sort of enhance sugar response and those things that would be very good for the future. So we're going very much into that now to make the food the new medicine. Health should come from the farm, not the pharmacy.
That's now you really left me off with wanting to go into another tangent and talk about this because I'm like, well, this is so interesting. So we might have to readdress this in the future because using food as a medicine makes a lot of sense. And now I'm really intrigued. But I feel like we could go on for a long time with this conversation. I learned a lot. You guys are both very smart on this topic, and thank you so much to the both of you for taking the time to chat with us today.
Okay. Thank you very much.
Yeah, thank you so much for inviting.
Yeah.
And thanks to everyone for tuning in. We really enjoyed having you here with us today. And if you missed part of this today's talk, please don't worry about it. You can check it out later on. It'll be available on demand on Facebook, YouTube, and LinkedIn. And if you have any questions about the subject or ideas for future topics that we should tackle, just feel free to reach out to myself or through the SeedWorld media channels. And next week, my colleague Mark Zinkwitz will be on to talk about with some guests about what role plant breeding has to play in a declining population. Yes, I said declining. So please tune in. Thanks for tuning in today, and make sure to tune in next week. Bye.